Lewis, the supplemental instructor for Dr. Byron Quinn's Natural Science Biology 1 course at Langston University, and I've compiled a series of review videos that will help you with your final. This is the next to the last one. This is Chapter 14. So in Chapter 14, we really focused on protein production again in RNA. So we're going to discuss proteins and amino acids, protein production, RNA versus DNA, base pairing, introns and exons, triplet code, the types of RNA, and codons. So a lot of this is a review, but we're just really kind of adding to it and going more into depth on some of these concepts. So again, you guys should know by now, and it will break my little heart if you guys don't remember that, but proteins are made up of amino acids. You guys know that. Please tell me you know that. Amino acids make up proteins. Proteins are made up of amino acids. You guys know that. We've been through that since the beginning of class. Okay, so just a review of protein production, and again, you guys should know this. The DNA transfers itself to transcribes itself to mRNA. The mRNA leaves the nucleus and goes to the ribosome, where it translates itself into a protein. You guys have learned this since, lots of chapters ago. So you guys know this pretty well. Okay, just a little bit more review, RNA versus DNA. We know that there are some differences between the two. Well, RNA, ribonucleic acid, DNA, deoxyribose nucleic acid. Well, one of the differences is the sugar. In RNA, you have the sugar ribose, whereas in DNA, you have the sugar deoxyribose. Another difference is the bases. In RNA, you have uracil instead of thymine. In DNA, of course, you have thymine. And then the third difference is that DNA tends to be double-stranded, where RNA is single-stranded. So just observe and write down those differences right now, and just take a moment too, if you need to pause the video, take what I just said in, and make your own notes so that you can remember that and study for your final. Okay, base pairing. We just went over this in the last chapter, but if we're going from DNA to RNA, just say this is our original sequence, GCAA, Well, our complementary RNA sequence would, of course, G's pair with C still, C's pair with G still. A's do not pair in RNA. There's no T's, so it pairs with a U instead. So that's why you see these U's here. C's pair with G's, A's pair with T's, A's pair with U's. C's pair with G's, C's pair with G's, T's pair with A's, G's pair with C's, and A's again pair with U's. So that's how, when we talk about DNA transcribes itself to mRNA, this is exactly what's happening. You have the original DNA sequence. It makes a copy of itself, mRNA, that can exit the nucleus. This is exactly how it does it. Alright, well then we talked about another concept called introns. Introns are what we call junk DNA. These are the portions of the DNA sequence that do not have a known purpose. However, this is a particular area of science that is growing because maybe, although the original purpose is not known, they're doing more research on these introns to see exactly what they do. So an example of an intron that I like to use is this particular example right here. You have a lot of what it looks like what we call nonsense, right? A lot of this doesn't make sense. However, the underlying portions do make sense. As a matter of fact, you have a little note for me that says, good luck on your final. Well, that is an example of the exons. So the exons are the part of DNA that makes sense. The part of DNA that makes sense, that have a known purpose. Well, the rest of this ACD, well, this doesn't make sense right here. This doesn't make sense. These would be our introns. So our introns is our portions of the DNA that don't make sense, or what we call our junk DNA. Well, what we can do is we can cut out the part that doesn't make sense with restriction enzymes. And when we do that, you have a clean part that just simply makes sense. Good luck on your final. So you can cut out the introns or the junk DNA with the restriction enzymes, and all you're left with is the exons. Well, the nucleotides, it takes 3 nucleotides to code for 1 amino acid, and we call this the triplet code. So if you see here in the DNA, we have G, C, and A, and the complementary mRNA sequence will be, of course, G pairs with C, C pairs with G, A pairs with U, because this is RNA in this case. Well, it takes 3 of these to make 1 amino acid. So these 3 particular mRNA nucleotides code for 1 amino acid, arginine, and so on and so forth. So 3 nucleosides to make one amino acid, and that is called the triplet code. Another concept is to remember that there are actually different types of RNA. We mainly focus on the mRNA because we know that it occurs in the nucleus. So the DNA transcribes itself to mRNA within the nucleus, and then it exits out of the nucleus to go function in the cytoplasm. Well, there's also another type of RNA, the tRNA, that's already in the cytoplasm. It provides that link between the mRNA and the amino acids. So it transfers them. It kind of acts as like the worker. It gets the amino acids and brings them to the ribosomes. The ribosomal RNA, or the rRNA, is already on the ribosome, and that is the main structural component of the ribosome. So that is the part that actually reads the message on the mRNA. The transfer RNA goes and gets the amino acids that the RNA calls for, that is encoded on the mRNA. If that made sense. If it didn't, rewind it, listen to it again. You might want to write that down. Alright, and last but not least, we briefly talked about start and stop codons. Know that there's one start codon, so this says, okay, now you can start making a protein here. And that is the AUG. That is our start codon. And there are 3 stop codons. You have UAA, UAG, and UGA. These are saying, okay, you can stop making a protein here. So that was quick, short, and sweet, and that covers chapter 14.